We are studying microorganisms. What are the microorganisms? Micro means very small and organisms means living beings. So what actually microorganisms are? Very small organisms to which we cannot see by naked eyes, which cannot be seen by naked eyes. So these are very small organisms which cannot be seen by naked eyes. So microorganisms are very small living beings. So the study of microorganisms, is called microbiology. It is microbiology. A micro means very small, bio means living, and logy means it is study. So what is microbiology? Means study of very small organisms. This study of microorganisms called microbiology. Now come to the next. It is how we can see microorganisms. So for the study of microorganisms we need to specific instruments. It is microscope. First, micro means very small and scope means to see. So what is microscope? Microscope is an instrument with the help of these we can see very small object to which we cannot see by naked eyes. It is microscope. So microscope is an instrument which helps in the study of microorganisms. So now come to the microscopes. First microscope was discovered or invented by two brothers. It is J. Jensen and F. Jensen. And the microscope that was invented by J. Jensen and F. Jensen was compound microscope. It was microscope. It was compound microscope. So now on the basis of microscopes are divided into two categories. Microscopes are divided into two categories on the basis of lenses, on the basis of lens number. When only single lens is used, this microscope is called simple microscope. And when two and more than two lenses are used, this is called compound microscope. So the first microscope which was invented by Jensen brother was compound microscope. They used two lenses. One was concave, one One lens was concave and another lens was convex. It is convex and it is concave lens. So the two lenses were first one was concave and second one was convex, two lenses. And come to the simple microscope, only one single lens is used. It is simple microscope. A scientist, Anthony von Leeuwenhoek, he invented simple microscope. That was simple microscope. With the help of this microscope, he could study. He could first study what it is, protozoans and bacteria, and he named this bacteria and protozoan as animalcule. What he said, animalcule to the both protozoans and bacteria. And the magnification power. What is magnification? It is to enlarge the object. Magnification power of the simple microscope, which was made by Anthony von Leeuwenhoek, that was 200 to 300 times. It is the sign of magnification power. So in this, the object can be enlarged up to 200 and to 300 times, so that Anthony von Leeuwenhoek can study protozoans and bacteria and name them animalcule. And another scientist, it was Robert Hooke. Robert Hooke, with his own made compound microscope, he could study cork cell. He studied cork cell and studied and named cell. So that cell name was given by Robert Hooke in his book Micrographia. The Micrographia book was written by Robert Hooke in 1665. Okay. Now the magnification power of Robert Hooke was 14 to 42. The magnification power of Robert Hooke's microscope was 40 to 42x. So these two microscope studies, one was simple and the second one was compound. Simple and compound microscope. Simple and compound microscope studied are not able to visualize very small. So in scanning electron microscope, we can study the surface, outer surface of an organelle, and with the help of transmission electronscope we can study internal structure of the cell we can study. So with the help of a scanning electron microscope we can easily study the cell membrane, and with the help of transmission electron microscope internal structure, ribosomes, microtubules, we can study ribosomes, microtubules. Very smallest organelles can be studied by the transmembrane electron microscope. Microtubules and mitochondria. Very smallest organelles can be also studied by the electron microscope and scanning electron microscope. Now come to the magnification power. It is about 15,000 to 20,000 x, or in case of transmission electron, its magnification power is 1 lakh to 4 lakh. It is magnification power. So in this electron microscope, transmission electron microscope, we can study object 1 lakh to 4,000 times 4 lakh times enlarged. In case of scanning electron microscope, we can enlarge object up to 15,000 to 20,000 x. Okay, now the microscopy studied electron microscope, and this electron beam is used so that cell cannot remain alive. The cell became dead. So the study of living cell can be done under special kind of electron microscope. It is phase contrast microscope. Study of living cell can be done in the another kind of very phase contrast microscope. So this microscope, phase contrast microscope, is used to study living cell. So the living activities, it is cell division and formation of sperms, spermatogenesis, oogenesis. It is formation of egg cells can be done with the help of phase contrast microscope. Now these are all about the microscope. Now we come to the type of organisms. The microorganisms are very small organisms. It is micron. It is 10 to the power minus 6 range. This is called micron size. So first, it is very smallest, it is viruses, then bacteria, protozoa, protozoans, then come to the algae and fungi. All organisms are considered in microorganisms: viruses, bacteria, protozoans, and algae, fungi. So first, what are the habitat? Habitat, this is the palace. What is habitat? Habitat is the palace where organisms can be found. Where organism can be found. So habitat for microorganisms. Microorganisms can present anywhere. Microorganisms can present anywhere. It is called omnipresent. They are universal. They are omnipresent. They are omnipresent. They can present anywhere. So example, they can be present in soil, air, they can present in water, and in very polar region, snow, and very cold environment, and very extreme haline condition. They can be present in very extreme haline condition. or it is saline condition. It is saline. They can be present in very harsh condition. Very harsh condition. They can tolerate very harsh conditions. Now come to the size of microorganisms. Size of microorganisms. They ranges from micron, 0.1 micron to of 0.5 micron maybe. It is the range of microorganisms, and they can tolerate very harsh condition. It is very high temperature, cold temperature, very cold temperature, means very less from temperature, very low temperature. They can present in saline condition. Now we will first take a bacteria. We will study first bacteria. So bacteria are prokaryotes. So here prokaryotes, pro means primitive or old, we can say old. Karyos means karyos means nucleus. So primitive nucleus is present in bacteria, means there is nuclear membrane is absent. In case of bacteria, nuclear membrane will absent in bacterial nucleus. Also this nucleus is called, bacterial nucleus will be called nucleoid. The nucleus of bacteria is called nucleoid. Now where they can present? First, bacteria may be present anywhere, means they are omnipresent. They can be present in air, soil, water, and its polar region, at the bottom of and at the very hot spring water. They can tolerate all harsh conditions. They can tolerate very harsh conditions. Now come to the size. They may be of various sizes. It is 0.1 to 0.6 micrometer range. And now shape. On the basis of their shape, they may be of various type. When it is spherical, the bacteria will be called coccus. It is singular. Coccus is singular, and cocci, it is plural. And when it is of rod shape, when it is of which shape? It is rod shape, then it will be called bacillus, and it is plural is bacilli. And there may be of spherical, there may be of spiral shape, and it may be of coma shape. It may be of coma shape. When two coccus are joined together, then the bacteria will be of diplococcus. And when two rod-shaped microorganisms are joined together, it is called diplobacillus. Okay. So example, it is lactobacillus. Lactobacillus is an enzyme bacteria. Lactobacillus, it is a bacteria which convert milk into curd. So the shape of this bacteria is bacillus. It is rod shape. So the lactobacillus is of red shape which converts milk into curd. Now first we come to the structure of bacteria. Now structure of bacteria. So we just take a diagram. It is the bacterial cell membrane, and through this bacterial membrane, in some microorganism there may be a outermost covering as well. It is capsule which is secreted by plasma membrane. So it is cell outermost it is capsule, and beneath this capsule there is cell wall. It is cell wall. It is made up of peptidoglycan. It is made up of peptidoglycan. Cell wall of bacteria is made up of peptidoglycan, and capsule is secreted by bacterial cell membrane. It is polysaccharides, and beneath this cell wall there is cell membrane is present. A cell membrane is universal phenomenon, and the invagination of cell membrane forms a structure. It is called mesosomes. And sometimes the cell membranes budding forms a separate vacuole in which some pigments are present, which helps in the photosynthesis. In case of bacteria, it is chromatophores. So it is the bacterial cell wall. Outermost covering is capsule. Then inside the capsule it is cell wall. It is made up of peptidoglycan, and it is green color. It is cell membrane. And invagination of cell membrane forms mesosomes. It is mesosomes. It is analogous to mitochondria. And what is the function of mitochondria? It is the respiration. It is analogous to the mitochondria, and mitochondria helps in the respiration. So the mitochondria helps in respiration. So in case of bacteria, the respiration will take place at mesosomes, and this is called chromatophores. This structure is called chromatophores. So this structure is called chromatophores. These are the chromatophores which helps in sometimes in photosynthesis. And bacteria, these chromatophores helps in photosynthesis. And there is its own genetic material is present. Its genetic material, it is double-stranded circular DNA. It is own genetic material is present. It is DNA. It is hereditary material, and there are ribosomes. Number of ribosomes are present, and the ribosomes helps in the protein synthesis. These are ribosomes which helps in protein synthesis. protein synthesis, and there is storage granules also present. What storage granules? Storage material. It is storage material. These are the storage materials. Okay.